The second results are we use network analysis method. So we have the functional connectivity matrix first, then we construct them into a network using graph theory approach. So these are the basic terms in network analysis. So a region, the 110 regions, each region is denoted as a node or a region, and the connectivity between regions are denoted as an edge. So this is a triangle, and this is a triplet. So the first network matrix we can use is about path length. So let's take this green node and the yellow node. So the shortest path length for them, for these nodes to reach each other, is through 3 edges. So the mean path length, the characteristic path length, or the shortest path length is 3. So the longer the path length, actually the less efficiency. So the reciprocal of this measure is actually called efficiency. So the high efficiency, that means you had better information processing or easier information processing between 2 nodes. The other measurement is about clustering coefficient. So it's all about the connection between the neighbors of a given node. Let's say this index node. So this index node, all the neighbors of it are connected to each other. So it had higher clustering coefficient. So it's all about how the index node's neighbor are connected to each other. So it's more about a local, functionally local connectivity measurements. So here are some network metrics, and the yellow ones are the ones that we measured in the study. If we want to measure functional segregation, then we can use the clustering coefficient or local efficiency, which means the efficiency of all the nodes immediately attached to an index node. So it's a larger measurement compared to clustering coefficient. And if we want to measure the functional integration about for a node to the whole network, then we're looking at its global efficiency. We also measure the centrality measures, that's the hubness, how important a node is. So the higher degree, that means the higher edges, the more edges that's connected to a node, the more central of a node. Between and centrality means how many shortest path lengths for the rest of the network needs to go by a node. So if that's the case, like Catania is a hub for Sicily or for Italy, then all the flights connected to the rest of Sicily Island needs to go by Catania. Then Catania is actually a hub. So it is the between and centrality measures the hubness of this region. Okay, so first we look at the global network properties. Surprisingly, we found no global differences of the network of people with autism or without. So no significant difference in small worldness, so how small world the network is, or global efficiency, or sortativity, and hierarchy. They are all the measurements about how the networks are constructed. However, there seems to be a marginal differences in clustering coefficient. That's lower clustering coefficients in people with autism. That implies that the network is kind of a bit random than the neurotypical networks. So this is a global measurement, but they're as efficient as each other. However, there are some regions that showed differences. So we have nodal differences, and these are the regions that pass false positive selection, so which passed multiple comparison. So for the red ones, it showed lower the regions like left anterior superior temporal gyrus and especially left posterior parahippocampal gyrus. They showed lower global efficiency and lower degree compared in people with autism compared to neurotypicals. And for centrality measures, middle cingulate cortex and again left posterior parahippocampal gyrus, they show decreased centrality in people with autism. And for clustering, we found posterior postcentral gyrus and the right anterior middle temporal gyrus. They show lower clustering coefficient in people with autism. And interestingly, in people with autism, there are 2 regions that showed higher degree and global efficiency, which is right nuclear accumbens and right posterior supramarginal gyrus. Okay, so here are our conclusions. First, we need to rethink about the theory of aberrant connectivity. When we're talking about connectivity, it's a complex term. It's not only about structural, it's not only about functional. We have to specify very clearly what kind of connectivity we're talking about: physical, functional, structural, or something else. And also, when we are talking about functional connectivity, we also need to rethink about the term, because what we measure in this specific research is resting state functional connectivity, which could be different from the task-induced functional connectivity. So there are plenty of evidences showing that people with autism, they may have decreased long-range connectivity in task conditions, but in a resting condition, tackling very low frequency oscillations, we cannot really find a striking global differences between the two groups. So this is the first thing we have to think about. And also, we cannot find striking network property differences. However, there might be a slightly towards randomness in people with autism. However, we do identify a region, that's orbitofrontal cortex, that seems to show lower connectivity in the resting state. And so this may help us rethink about the global versus local debate. When we are talking about network, we always want to talk about a lot of regions. But when we really tackle the problem using a whole brain approach, then we cannot find a whole brain difference. We can only identify a local difference again. So this is quite tricky. But I think this is one of the important messages, that we have to think about local properties again. And OFC is the region that we found. And also, apart from OFC, there are other regions that may show differences between groups. So there might be a shift of regional specificity in autism spectrum conditions. For example, we have very hypoconnected orbitofrontal cortex, and the left posterior parahippocampal gyrus is much less central, it's much less important. And we have less segregation around the primary sensory cortex, and we have higher efficiency in right nuclear accumbens and posterior supramarginal gyrus in autism. That may give us some implication of the information processing process in people with autism. But lastly, we do have some limitations. First, for all the network analysis, how you parse the brain may bring you different results. Also, using different postulation scheme, you may have similar global comparisons, but the nodal comparisons would be different. So we have to keep in mind that this is a postulation based on 110 nodes. But if you use another scheme like 65 nodes or 200 or 500 nodes, that may give you different results. Secondly, the neurobiological interpretation of all the network metrics should be carefully studied in the future. The study is supported by the United Kingdom Medical Research Council AIMS Consortium, which is a collaboration between University of Cambridge, Institute of Psychiatry in London, and University of Oxford. And it is supported by Medical Research Council UK, and my personal financial support is coming from Ministry of Education Taiwan. And we have to thank all our research volunteers. Thank you very much.